I'm Brenda Hemmelgarn, Dean of the Faculty of Medicine and Dentistry, and very pleased to be your MC for this event. Thank you all for joining us today for this very special celebration. Even though we can't gather in person, I'm so pleased you've all been able to make the time to be here virtually. Before we continue, I want to acknowledge that we are located on Treaty 6 territory, and we respect the histories, language, and cultures of First Nations, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. As you all know, in early October, the faculty received the wonderful news that our very own Dr. Michael Houghton had been awarded the Nobel Prize in Physiology or Medicine, which he shares proudly with Harvey Alter and Charlie Rice. The decades-long work by all three in the area of hepatitis C has changed the lives of millions. I'll give you a little bit of background. In 1989, after 7 years of research at an American pharmaceutical company, Dr. Houghton and his colleagues, Qiulin Chu and George Koh, unlocked the genetic code of the hepatitis C virus. This discovery has since opened a variety of research paths leading to improved blood safety and a level of treatment that ensures the viral infection can now be cured in nearly all patients. It also started Dr. Houghton on a path of his own that eventually led him here to the University of Alberta, where he was recruited to the position of Canada Excellence Research Chair in Virology in the Shing Institute of Virology in 2010. He spent the majority of the 10 years since working on a vaccine, which is now in the late stage of preclinical testing. These days, Dr. Houghton is one of the countless researchers across the globe hard at work on tackling COVID-19, building on his discovery of a vaccine for SARS in 2004. A few common themes emerge in everything that Dr. Houghton does. The field of medical research is vibrant and constantly changing, and I share with Dr. Houghton the passionate belief that the best advances in this area are made through collaboration and interdisciplinarity. Scientific discoveries do not happen in solitude. Science is all about teamwork. And today, more than ever, we see the truth in this, as all over the world, experts are coming together every day to fight the global coronavirus pandemic. Solutions to problems as complex and challenging as this can only be found when different departments, faculties, and schools collaborate rather than compete. And when we break down barriers between states and nations and pool our intellectual and financial resources for success. Advances like Dr. Houghton's also take time and patience. And as he himself recently quoted as saying, there is no one eureka moment. Vaccines and treatments for the world's most common diseases do not happen overnight. They involve long hours in the lab and months of analysis, sometimes years of testing and retesting, so that in the end, the communities we all aim to serve will have fair and easy access to the best possible solutions to their health needs. I want to thank you all for your enthusiastic response to our request for questions for Dr. Houghton today. We're so grateful for your participation. Since we only have an hour, we've narrowed down the list to a handful of the most popular questions submitted. And I will look forward to discussing those with Dr. Houghton a little bit later in the program. But don't worry, if you didn't get a chance to submit a question ahead of time, there will be time near the end of the program for a brief Q&A with Dr. Houghton. Today, we're very fortunate to hear not just from Dr. Houghton himself, but also from our very special guest, Dr. Lauren Tyrell. Dr. Tyrell is a distinguished University of Alberta professor, a virology researcher, and the founding director of the Shing Institute of Virology. He's here today to share a few words about his close friend and colleague, Dr. Houghton. So please join me in welcoming Dr. Lauren Tyrell.
Thank you, Dean Hemmelgarn, for your kind introduction and for organizing this event to recognize Michael Houghton's winning of the Nobel Prize in Medicine for 2020. I have known of Michael Houghton since he led a team at Chiron to discover the hepatitis C virus. However, I got to know Michael Houghton in the early 2000s, and particularly since he was recruited to the Shing Institute of Virology and the University of Alberta in 2010. Michael is a man of many interests and talents. He's an obsessive reader, an avid sports fan, particularly for cricket and soccer, a compassionate man with a broad interest from politics to social injustices. Most of all, he's a brilliant, passionate, and focused scientist, a wonderful collaborator who understands the power of multidisciplinary research and teamwork. He is an inspiring colleague. Non-A, non-B hepatitis was a huge problem in medicine in the 1980s and was clearly associated with blood transfusions. Between 1974 and 1989, many labs tried to find this virus and failed. Michael's vision and passion and 7 years of hard work led his team to discover the virus in 1989. They also developed a test to detect the virus in blood. In fact, I will show you from the first vials of the reagent produced by Chiron that Michael and his wife Ida gave me on one of the visits to their home. It is on display in the Li Ka-shing Institute. Canadians will know how important this is, as 30,000 Canadians were infected by blood transfusions, and this was the subject of the Creever inquiry. Michael's discovery has saved millions of lives worldwide. Every year for the last 8 years, I've supported the nomination of Michael Houghton and his two colleagues, Quilim Chu and George Koo, for the Nobel Prize in Physiology or Medicine. Nobel Prize candidates can be nominated by previous Nobel laureates, selected institutions, and on a rotational basis by individuals. I want to thank Michel Chrétien, the brother of former Prime Minister John Chrétien, for his strong belief in Michael and his colleagues, and that they were excellent candidates for the Nobel Prize, and he supported the nomination with me for the last 3 years. There have been many great researchers working on hepatitis C, and certainly Harvey Alter and Charlie Rice were deserving winners with Michael. A number of scientists could have been chosen. However, in my mind, any combination you put together always had to include Michael Houghton. I believe when Michael was recruited to the Li Ka-shing Institute and the University of Alberta, Michael would one day win a Nobel Prize in medicine. I had someone call me from the UK who was writing a book on people who should have been awarded the Nobel Prize but were bypassed. He wanted information on Michael, and I refused to give him the information simply because I believed that Michael would still win a Nobel Prize. Michael was recruited to the University of Alberta based on the generosity of the Li Ka-shing Foundation with $28 million and the Alberta government to establish the institute. Michael was in the first round of Canada Excellence Research Shares in 2010, a program designed to attract outstanding scientists to help initiatives like ours in the Li Ka-shing Institute to be competitive internationally, and Michael certainly achieved that goal. There were key people who supported the effort to recruit Michael, including David Evans, the chair of MMI, Lauren Babiak, the vice president of research, Indira Samasareka, the president of the University of Alberta, and from the government of Alberta, Premier Ed Stelmach, Annette Trimby, and Dave Hancock. And from the Li Ka-shing Foundation, Selena Chow, Julia Sao, and Frida Law. And finally, I want to mention John Chu from the University of Alberta, who opened many doors and guided the cultural interactions that were so critical to the success of this initiative. Most importantly, Mike, who is a visionary and passionate scientist, has always stressed the importance of research benefiting patients. When Michael met Mr. Lee, this resonated between the two of them, as Mr. Lee had stressed the desire to see research in the Institute to bring benefits to patients. Finally, while believing Michael and knowing that the Nobel Prize in Physiology and Medicine is the first of the Nobel Prize awarded each year and always on the first Monday in October, I woke up a few minutes before 4 a.m. on October the 5th and checked my iPhone. At first there was nothing, and suddenly it appeared that Michael had won the Nobel Prize with Harvey Alter and Charlie Rice. I waited a few minutes and then I called Michael. He asked if everything was okay, and I said, "It's wonderful. You've just won the Nobel Prize." I thought he would have been called by the Nobel Committee at this time, since it was already public, but they had not reached him. We shared that wonderful moment and what it meant for Michael to be finally recognized, what it meant for the University of Alberta, for Alberta and Canada. This is only the second Nobel Prize in Physiology or Medicine to come to Canada, the first being to Benton and MacLeod in 1923 for the discovery of insulin. After nearly a century, Canada has its second. Michael always believed the only way to solve difficult problems is through research. Currently, a small piece of RNA, the coronavirus, has brought the world to its knees. Michael has firmly believes that research will solve our current problem of COVID-19, and he has worked on this as well. Thank you, Michael, for all of your hard work and your passion for discovery. You have transformed the Faculty of Medicine and Dentistry. You have transformed the University of Alberta, and you have transformed the world. Congratulations, Michael, on behalf of all of us at the University of Alberta.
Thank you very much, Dr. Tyrrell, for that amazing tribute. And thank you as well for your insight in recruiting Michael here to the University of Alberta and your support of the nomination. What I'd like to do now is actually ask everyone to join me in welcoming the Nobel Prize winner himself, Dr. Michael Houghton.
Thank you very much, Dr. Hemmelgarn and Dr. Lauren Tyrell. Those were wonderful tributes, which I very much appreciated. Thank you. Thank you also to your colleagues throughout the university for all of your many congratulations, and also for the arrangements around the Nobel Prize. Well, as Lorne mentioned, I was woken up 3 o'clock in the morning, my time, and I thought, well, maybe it is the Stockholm phone call at last, but in a way it was more fitting to actually find out it was Lauren Tyrell on the end of the phone, who has continuously supported me on all things while I've been at the University of Alberta. And as Lauren said, he said, "You got it, Mike." And I said, "Well, I haven't received a phone call from Stockholm yet, Lauren, so it cannot be." He said, "It is." So a couple of hours later, I did get the call from Stockholm. So it was a wonderful moment. Wonderful moment to tell my wife also. That was fantastic. So I think a lot of people are asking me what it's like to get a Nobel Prize. And of course, it is tremendously, one is tremendously honored when one gets a Nobel. There's no prize quite like it in the whole world. And it's been running for hundreds of years due to the courtesy and generosity of Alfred Nobel. I thought it would open doors, but I've been really shocked just how many doors it does open, and especially through emails that we all use today. The number of emails for the last 2 months has just been incredible, and it's very nice. Lots of student groups want me to talk, and lots of other organizations, and it has been very nice and an opportunity for me to give back at different levels to the Nobel. I think, as Lorne implied, getting these big awards is a little bit bittersweet, very sweet for the prestige and for the honor, but a little bitter that, or slightly bitter that none of my close collaborators who worked with me to discover the Hep C virus were directly honored. And they are Dr. George Kuo, Dr. Quilim Chu, who worked with me at the pharmaceutical company I worked at in discovering the virus, called Chiron in California, and also Daniel Bradley at the Centers for Disease Control, who supplied us with animal samples continuously throughout the 7 years of trying to find the virus. So I very much have those people on my mind these days, and thank them, of course. Without their input, I would not be talking to you today. People have asked me, well, where do I go now with the Nobel? Well, as Lorne said, when I was 17, I read a book about Louis Pasteur, and I read about his work, obviously, and I read about the circumstance where his daughter died of a bacterial infection, and he dedicated himself to trying to stop that happening to other people. So we have plenty left to do on HCV. We have very good diagnostics and very good blood tests. We have now very good antivirals thanks to the combined efforts of academia, the pharmaceutical companies, public institutions like NIH, and public health agencies. We now have very good drugs to cure most people very quickly with minimal side effects. So that's been a big breakthrough. And I believe a vaccine is needed to effectively control the epidemic. And for the last 10 years at the U of A, working with a great team of young scientists, I think we're on the path to delivering a vaccine. And I think one thing COVID has taught us is that maybe we can speed up the development in certain ways. So I expect the vaccine for Hep C to become available over the next 5 to 7 years, hopefully faster. And as Lorne said, my goal in my career has been to make a contribution to preventing disease. I never had ambitions for big awards like Nobel or some of the other awards I've won. My ambition has always been to make a contribution, doesn't matter how small, to preventing disease. Unfortunately, there are so many diseases that afflict mankind, and HCV is just one of them. And working with Lorne and our collaborators and our teams in the Shing Applied Virology Institute, we are addressing many serious diseases like Group A Streptococcus, which causes heart disease, rheumatic heart disease, Alzheimer's with Professor Jack Germandes, cytomegalovirus with Dr. Jim Neiman, who's in our institute, fatty liver disease, which now becomes one of the major problems in hepatology, as well as many other collaborators working on anti-cancer strategies. So basically our goal, along with many people listening today to this webinar, is to prevent disease. And there's still a lot left, so we just keep on. HCV has been a success so far, but there's plenty of other diseases that need our attention. And so thank you again to Dr. Hemmelgarn and Lorne. Lorne has been the most amazing colleague for the last 10 years, where I've been working with him closely. He's a phenomenon, and it's been an honor for me to work with him. And thank you again for attending this presentation.
Thank you very much, Dr. Houghton. I can only imagine what your inbox was like after that announcement went out. Inundated would be something to say it in a minor way. And what an amazing career goal, to make a contribution to prevent disease, and it doesn't matter how small that is. It's something I think we can all aspire to. So thank you for those words of wisdom. What I'm going to do now is we actually invited individuals to send questions in that they would like to ask a Nobel laureate. And we compiled those questions, and I've got a few of them here. So I'm going to ask you some of these questions that have been submitted to us. And the first one speaks to something that we as researchers often face, and that's some of the challenges and some of the frustrations with unsuccessful grant applications, for example. And the question is, tell us what challenges you've had to overcome in your career.
Well, as as I mentioned, and also Lorne, the discovery of the Hep C virus was done in a pharmaceutical company. It was a startup biotech company formed by a couple of University of California professors, Dr. Bill Rutter and Dr. Ed Penhote. I think in a biotech environment, you're obviously trying to innovate in medical research. Investors give you money and they're assuming that at some point you will be successful, at least on one project in the company. So Hep C, I knew was going to be difficult to identify. I was a molecular biologist, trained molecular biologist in the United Kingdom before I went to Chiron in California. And even though we didn't have a molecular handle for the virus, it really was looking for a needle in a haystack. I knew it was going to take a long time. So the pressure really was, it lasted 6-7 years before the world accepted that we had identified it. And there are pressures when you're in industry to deliver. But at the same time, I wasn't worried too much because deep down I felt we would do it. And secondly, I felt, well, what better project could I be working on to try to identify a virus that was afflicting so many millions of people around the world? So I think that was probably the situation, Dr. Hemmelgarn. I felt instinctively that we could do this somehow, and that's what kept me going. And I think the importance of the project kept me going as well.
And the importance of determination. You found your needle in your haystack. The next question that we have relates to COVID-19, actually, which isn't surprising. And the question is, what lessons from hepatitis C do you think apply to COVID-19 and vice versa?
Yes, that's a question I've been asked a lot about recently. And 2 months ago, it was exactly the question you posed. How can HCV teach us about COVID? But now, I think the reverse question is applying. What can COVID teach us in HCV and in other infectious diseases? So I think the global response to COVID has been terrific. I think the way academics have worked with corporations, have worked with public health organizations and governments, it's been a tremendously coordinated effort. And I think that has been part of the rapid progress with COVID vaccines. And in addition, we've seen new technologies applied to COVID that had never been applied before for other vaccines. And I'm talking obviously about the RNA vaccine technology and also the adenovirus-based technology. So both of those technologies really are quite recent. Adeno was developed by Dr. Adrian Hill at Oxford University for preventing malaria. But he and his team and Dr. Gilbert quickly applied it to COVID. There has never been a vaccine approved using that technology. And of course, the RNA vaccine technology is even newer. The first paper that I have seen from that technology was literally 3 years ago in the context of influenza. So I think now we have very good news on the vaccine efficacies so far using adeno and RNA vaccines. We hope that the protection will last for many months, if not 1 or 2 years, if not longer. But we're still waiting to see the long-term protection. But I'm extremely admirable. I have great admiration for the people that have developed these vaccines, the way everyone's worked together. And indeed, we are now looking at applying RNA vaccine technology to hepatitis C. And in theory, the technology of RNA vaccines can be applied to pretty much all vaccines, be they viral or bacterial. So I think we've learned from COVID, and I think we've learned how to work together as well at all levels of society to really speed up vaccine development. So I think that will have repercussions as well for other vaccines, including hepatitis C. We can do it faster than the traditional timeline, which has been around 8 to 10 years to get a vaccine approved. Now, hopefully, the regulators will approve emergency use of these new COVID vaccines. And that, if it happens, it will be within 1 year, as you know. So that's unheard of for vaccine approval. So I think I'm looking forward to applying the technologies of COVID to Hep C and also to many other vaccines in the future.
Yeah, as challenging as this pandemic has been, there will be positive outcomes. And I think the efficiency in terms of vaccine development and getting it to market is one of those things. And the coordinated effort that you spoke about as well, and the collaborations, I think we've learned a lot from that. Related to that, when you spoke about the coordinated effort, the next question actually asks specifically about emerging pathogens, and it says, what should we be doing more or less to be ready for emerging pathogens?
Yes, I think one of the most sobering conclusions from the COVID epidemic is that if we had stockpiled vaccines against the SARS epidemic in 2003, those vaccines could have prevented a lot of the disease amongst the human community from COVID. We know that vaccines to the SARS 2003 strain of the coronavirus can actually and do actually cross-neutralize the infectivity of COVID. And what happened, because the SARS 2003 epidemic came quickly, everybody focused on it, vaccines were researched and got to the point of manufacturing, but then the epidemic declined because of public health measures and manufacturing stopped. And so I think this is a big lesson from SARS 2003 and now COVID. We must stockpile vaccines to these pandemic strains because in the future, we can be sure there will be other pandemics, maybe coronaviruses, quite possibly influenzas. And I think that is a clear lesson. We must stockpile vaccines to strains that emerge because there is a decent chance that they will be effective, at least partially, against new strains that emerge. I think the other lessons from COVID are, it's told everyone in the whole world how vulnerable we are to infectious disease. And so maybe a few decades ago, humans became confident that we were masters of our universe. We knew how to deal with infectious disease, but clearly we don't in terms of being vulnerable to new epidemics. So I think this is a wake-up call also for funding agencies. We need to increase funding for basic research as well as translational research in infectious disease. And I think also another lesson from COVID for us in Canada is that we've realized we don't have the manufacturing capabilities that we really need to have some independence from other countries making vaccines for us. So there are some really important lessons that we must, and I'm sure we will learn from COVID.
Very important points. The importance of stockpiling vaccines and the vulnerability that we have to infectious diseases. I think we can all just think back to John Snow and that pump handle decades ago, and that is just a replication here today with COVID. I'm going to take you away a little bit from the science now and more of a local type question. And this next question says, what has the U of A been able to offer you that has been critical to your team's work and success this past decade?
Yes, well, I've been at the U of A 10 years, as you say, and it's been certainly one of the most, if not the most stimulating parts of my career. And I think that's for many reasons. One is collaborating with Lauren Tyrrell has been very special. We're very good friends. Haven't quite got to the point where I'm as ardent a fan of the Oilers as he is, but I'm sure that will come. But fundamentally, Lorne and I think similarly. We're both really focused on addressing unmet medical needs and doing something about disease. So, we have a lot of commonalities in that regard. I haven't quite persuaded him to enjoy cricket yet, but hopefully that will happen. I think the other great thing at the U of A is I've met and worked with so many stimulating professors and their labs across the campus. Many, of course, within the Faculty of Medicine. Obviously, what I try to do and contribute to requires interactions with medical doctors. They tell me about the disease that people have. They tell me how it develops, how it interacts with the host. And that's really important for someone like me trying to intervene. And of course, we get access to a lot of samples from patients with different diseases, and that's critical for researchers like me. But also very somewhat surprisingly, but very, very nicely, I've been able to collaborate with people like Professor Jack Tuzinski in the physics department and the oncology department on computational drug discovery. He's been a pioneer in this field, and he's one of the leaders in the world. And so we've really applied ourselves to his science, and a lot of his trainees have worked with us in my Circ chair lab originally, but subsequently also in the Shing Applied Virology Institute. So it is definitely a new era of drug discovery, and computation will play an ever important role in that process. It's been a pleasure to work with Jack Tuzinski. Professor Fred West in chemistry has supported us very well on many programs in synthetic chemistry. We've had some wonderful collaborations with pharmacy. Dr. Khalid Barakat is a professor in pharmacy, and we're working with him on trying to make small molecule inhibitors of the immune checkpoints, which have revolutionized cancer therapy, especially in melanoma, metastatic melanoma and lung cancer. And also, I've worked with Professor Sergei Noskov down at University of Calgary, as well as Dr. Carla Coffin, a hepatologist who has been working with Lorne for many years. And we've really enjoyed interacting with the people in Calgary as well. And I think there really are some fantastic and very cooperative minds in Alberta. And that's been a real positive for me. And it's been really so nice to interact with so many deep minds, if you will. I need to, the great time I've had at the U of A has been made possible by the federal government, who started the Canada Excellence in Research Chair Program in 2010. Indira Samarasekera, our ex-president, was instrumental in starting that program across Canada. So thank you to Indira, who's played a major role in initiating the CERC program. As Lorne said, we've had really tremendous support and enthusiasm from the Li Ka-shing Foundation, from Mr. Li Ka-shing and Selina Chow and their colleagues. They've donated to us substantially, and we've been able to use that funding along with other fundings to really get to grips with many, many serious diseases. And of course, the government of Alberta has really also been tremendously good to us. They've given our Applied Virology Institute, the Li Ka-shing Applied Virology Institute, substantial funding over the last 10 years. So a big thank you to all of our sponsors. And University of Alberta is really a great place to work as a researcher. I really have enjoyed it and look forward to continuing it. There's a lot of great minds there to work with.
You said it so well, Michael. It's all about the people. People are our greatest assets, and supported by great funders and partners. And
I'm sorry, Brenda, I had another couple of points, which was my fault. Sorry to interrupt you, but I do want to thank, I had a team of young scientists in my CERC lab, many of whom have joined the Shing Applied Virology Institute. So we have very talented young scientists, many of whom were trained at the University of Alberta. And the list is long, so I won't attempt to name them, but we really do have a talented and well-trained young scientist core that will guide us in the future going forward. And I'd like to thank, we have tremendous facilities at the U of A. I mean, the CATS Health Research Building is a state-of-the-art modern laboratory facility, and Lorne and many others were involved in putting that up. We have the beautiful Shing Applied Virology Institute facility, state-of-the-art labs where we can do our work, and incredibly, which is really quite unique, not just in Canada, but through the whole of North America, our Applied Virology Institute is next door to the Alberta Cell Therapy Manufacturing Unit run by Greg Corbett, and we can prepare vaccines and therapeutics in there and other biologicals for clinical trials. So that's a very unique set of facilities we have at U of A, which really helps us to be at the forefront of treating disease and preventing disease. Thank you, Brenda. Sorry for the interruption.
No. Great people, great facilities, and a great place to work. You said it all so well. The final question from this group of submitted questions will now take us more to the city of Edmonton and the Edmonton region. The question is, the city of Edmonton has set its sights and strategy on connecting, promoting, catalyzing, and accelerating the healthcare innovation ecosystem in the Edmonton region. And the question is, how important are the University of Alberta and the post-secondary sector in that health city effort? And the second part of that is, what has to be done to continue to attract and retain biomedical talent here?
Mm-hmm. Well, I think it's great that Edmonton has initiated its Health City initiative. We would be very happy to join with them. We talked to Minister Schweitzer fairly recently, and he's very enthusiastic about Health City and about how the U of A can contribute in making Edmonton a center of excellence for research and delivery. I think in order to, I think U of A attracts many talented people and retains them. I think going forward, once again, we go back to COVID. I think COVID has taught us that governments need to increase funding to infectious disease research, virology, bacteriology, immunology, and related fields, not only at the basic level, but also the translational level. So I think the response to COVID has been good scientifically, but I think we can do better. We need to increase our research funding and our translational funding, I believe. And we need to improve our manufacturing capabilities. COVID has clearly shown we are vulnerable to viral infections. The next one could be a pandemic influenza, or it could be a particularly bad drug-resistant bacterium. We've had those before, and we're probably going to have them in the future. So I think really we need to increase funding. We, being the Canadian community with government support, we have to increase funding in infectious disease research, I believe.
Thank you very much, Michael. Totally agree. Funding is just, it's essential to what we do, and we require that support. Fully agree. You've spoken frequently about the importance of trainees and training the next generation, and you just spoke about young scientists being core to your program. So what we've done now is we've invited 3 students actually to come forward and ask questions that they may have for you. So I'm going to invite first off Katie Bedore, who's a postdoctoral fellow from the Department of Biochemistry, to please ask a question.
Hi, I'd like to just start with thanks so much for taking your time today to speak with everyone. I know me and all of my colleagues have been really inspired by your news. So my question is, what's your best advice for the young investigators and trainees in the audience who are just embarking on their research careers?
Thank you, Katie. You know, one thing I haven't mentioned so far is when we worked in the 1980s, obviously more than 30 years ago, on trying to identify Hep C, that was before the advent of polymerase chain reaction. We did not have that ability to amplify the minute amounts of viral RNA. So it was very difficult to work with. I remember I made 8 cDNA libraries from different infectious animal samples. Only one had hepatitis C sequences in it. So, and then we didn't have the benefit of deep sequencing. So basically what took us 7 years in the 1980s probably would take 7 weeks now. And that's because we have these great new technologies. And, you know, the Nobel in chemistry was awarded to the CRISPR inventors. And you know full well how important CRISPR is to what we do in the lab. So my advice is make sure you keep up to date on new technologies, absolutely vital. And as our knowledge is growing exponentially, so is the number of technologies that we can all use in the laboratory. So pay attention to what other people are doing and how their technologies can help you. You know, the method that we finally used to identify Hep C, expression cDNA screening, that was a process that was developed by a couple of scientists at Stanford University in 1983. And eventually, after a lot of work, we got that to identify Hep C. So, you know, I would recommend every student, every trainee spending a morning looking at what new technologies came out this week and seeing if they can help you. I think also, you know, to train ourselves in virology, immunology, bacteriology, we have to be able to do work on soluble questions, how the viruses and the pathogens work in the cell and so forth. So you need to work on systems that you can study, that you can ask questions of and give data, make hypotheses and test the hypotheses. And that's how we train ourselves. And obviously that is still essential. But I think it would be, I would recommend at some point in your careers, and maybe fairly early on, even as a postdoc, spend some of your time thinking and maybe doing some work on unmet medical need, on serious diseases for which we know very little. Can't be 100% of your time because there's a risk you may not get a job afterwards without papers, as you well know. But I would advocate all young scientists spend some of their time, you know, maybe 20%, on considering how you could approach a severe disease for which we have very little information about. I think then you're doing a combination of great molecular science with your other projects, and you may be able to come up with a career in a disease that perhaps you can solve or help to solve. Thank you for your question, Katie.
Thank you so much.
Great question. Thank you, Katie. Now I'm going to invite Farah Elawar, a PhD candidate from the Department of Medical Microbiology and Immunology, to ask that question.
Hi, Dr. Houghton. Thank you for your advice and your inspiring words. It's a real honor to be in the MMI department. So persistence seems to be a personal and team quality of great importance to you. I'm wondering what you see the value of persistence is for individuals, families, businesses, and government, especially in this time of COVID right now.
Yeah, thanks for the nice question. You know, thinking about your question, I quickly realized persistence is a strong trait of human beings, right? I think our history and our successful history is largely due to persistence, whether it's at the level of Alberta pioneers, at the level of indigenous peoples surviving in difficult climates, and lots of other things, of course. you know, my own family, grandparents and so forth lived through, as millions of others did all over the world, lived through World War II. So persistence is in our genes for sure. And persistence counts a lot, I think, in all aspects of our lives. Unfortunately, being human means we sometimes have disappointments. They could be a failed experiment, but relative to other disappointments where we might lose loved ones, for example, their lab problems are relatively minor. So I think we're designed to persist. I think we've evolved to persist, and I think sometimes it gets tough. But in my experience, persistence pays off because eventually things work, and also personal matters. You know, we all have challenges in our personal lives, and I think it's important to work through them. They will get better, and they do make us stronger. If we have a setback, getting through that and surviving makes us stronger. So I think we have to persevere. And I think for me, that's a big part of my success, being persistent. I think I'm a little like a dog with its bone. I won't give up. Of course, you have to be realistic. I should not have been persistent at getting on a spaceship to Mars, because I know I couldn't have done that. I get giddy on the 7th floor of the Shing Applied Virology Institute. So you have to obviously ameliorate your persistence with a knowledge that eventually you will be successful, or a knowledge that things will get better in your personal life. So just keep going. I think that's my message. And in terms of science, I just want to conclude that my success is largely based on working with good colleagues, good scientific colleagues. I think in part it's based on my passion for solving disease. After I read the wonderful book about Louis Pasteur when I was 17, and persistence. You haven't failed until you quit. So thank you for
Yeah, thank you for your wonderful advice.
Passion and persistence, so well said. And now I'll invite Stephanie Zaney, who's a PhD student in the Department of Physiology, for their question.
Hi, first of all, congratulations, Dr. Houghton. And I have a double question for you. So what insights can you share on the overall value of scientific evidence to society? And how can we better advocate for the importance of basic science?
Yes, unfortunately, yeah, thanks very much for the question, Stefan. As you know, we are living in an age where science is questioned. It's very unfortunate. I think, you know, it seems to me it started with global warming, where scientists were being questioned about the accuracies of the effects of global warming. And now, of course, we've seen in many countries opposition to accepting the science of COVID. So I thought about your question for some while, and I think the answer is, and by the way, you know, the university, when I joined the University of Alberta in 2010, they agreed that I could be part-time in Edmonton and part-time with my family in United States, in California. So I have some friends who, shall we say, have different opinions about COVID and global warming to what I have. And I think the way to approach them is really, we as scientists, we have to present our case persuasively and helpfully. And I think we have to reassure people. I think a lot of people get scared, understandably, when you see a pandemic like this, and when they're told, well, your house will be underwater in 50 years' time, you know, so you better plan ahead. I think there's a fear factor that causes resistance to science. And I think we as scientists need to respect that fear, and we need to present our case persuasively and helpfully. And it doesn't help if we are sort of accusatory or critical in to people that do not accept science. So I think what I try to do is I try to illuminate people's minds who are not scientists and who are worried about various things, and I try to reassure them. And I think that's my approach, and I think we're going to need to do that as well, because the COVID vaccine will become available soon, and we need to reassure people that if our regulators really find people like Health Canada think it's safe, they've pored over all the data given to them by the companies, including efficacy and safety data. If they consider it safe enough to use, then we need to reassure our citizens that they should take it for their own good and also other people's. So I think we need to do it in a very positive, careful way, basically. So it's a great question. Thank you.
Thank you.
Stephanie, the importance of educating others. We do have a few minutes left, so we have the opportunity for one or two additional questions if anyone would like to ask Dr. Houghton a question. So please just raise your hand, or Emily will help assist in that. We do have one question in the chat box here I could read off. So is a former student who, in biochemistry at the U of A, who helped develop an antifungal vaccine against Candida. What do you think about the public perception of vaccines and people who don't believe in the power of vaccines, especially now that the entire world is waiting for the COVID-19 vaccine?
Yes, it's a great question, obviously. Well, vaccines really are, if you had to pick an area of human health where the greatest benefit was provided for the minimum risk, it would have to be vaccines. You know, tuberculosis, polio, childhood diseases, diphtheria, and so forth. Really, there's nothing as effective as the vaccine area. They keep us healthy, they keep our children healthy. The benefit to risk is absolutely hugely in favor of benefit. And so, once again, we just need to convince and reassure our citizens who are non-scientists of the great benefits of vaccination, not just to themselves, but, you know, I can think of only one thing worse than dying from COVID, and that is if you transmit it to another person who dies because you were not vaccinated. So Health Canada will review the data with a fine tooth comb, and if they say it's safe enough to use as emergency use, everybody should take it when they have the opportunity.
Thank you. Question from Paige. She says, I was just curious, why was it so difficult to isolate hepatitis C?
Well, Paige, I would advise you to listen to my Nobel lecture on December the 7th, which I believe is available on nobel.org. So I give a 30-minute lecture on why it was so difficult. I think in a nutshell, as I mentioned already, we didn't have the benefits of polymerase chain reaction. We didn't have deep sequencing. We didn't have a molecular handle for Hep C. So we were using quite crude methods of using radioactive cDNA probes from infectious samples and control samples, looking for a signal in one and not in the controls, obviously. The literature in that time was filled with evidence that non-A, non-B hepatitis, as it was then called, was related to hepatitis B, hepatitis A, yellow fever, bovine viral diarrhea virus. So we used all those hybridization probes without success, and that's because Hep C is a unique virus. We now know that. We tried hard to grow it in culture. You know, that was the breakthrough for HIV. The group at the Pasteur showed that they could grow the virus and actually see the retroviral particles in the live cells. We looked for antigens and antibodies associated with non-A non-B. That was the breakthrough for hepatitis B by Dr. Bruce Blumberg when he discovered what's called the Australia antigen in Australian Aboriginal peoples. He didn't know what it was. It turned out to be the viral envelope protein of Hep B. So we couldn't find any of those markers for non-A non-B. And so eventually my colleagues, Dr. Kuo and Dr. Chu and myself, we used cDNA expression. So we essentially made proteomic libraries derived from all the nucleic acids in Dr. Bradley's infectious plasma. And then we screened it with sera from patients, assuming the antibodies were there, even though nobody had shown they were there. And then, of course, we got quite a few positive clones where the antibody was binding to its target antigen. And like I mentioned, out of 8 libraries, eventually one of our libraries had one clone that we could show was derived from Hep C. And it was a tiny clone. It only had 100 base pairs in it. And that was it. That was the key to Hep C, good old 511 clone. The whole Hep C field was developed from that one clone. So it's not easy to do it. Actually, I would, if you have a spare moment in the lab, you could try it. Try finding it just using expression screening and patient sera. It's not easy. Without using PCR.
Sounds like a real challenge. We look forward to your lecture next week to understand more.
We can take a live question. I'm going to unmute Len and he can unmute himself. Len? Unmute yourself. Okay, we'll move along to Catherine. Please unmute yourself.
Hi there, thank you. So thank you for a tremendously insightful and beautifully humble discourse. And thank you, Brenda, for offering this opportunity for us all to interface with Dr. Houghton. So my question is this, and it may be a dumb question. Do you see a role for antibody testing, such as persistent antibodies? You mentioned the potential cross-reactivity, even with SARS, in COVID vaccination programs, for example, in deprioritizing those who might have antibodies and might not need the vaccine as much as others who might not?
Yeah, very good question, Catherine. I hadn't really thought about that before. Yeah, as I said, there is cross-reaction at the antibody level in the receptor binding domain of the spike proteins of COVID and SARS 2003, as I like to refer to it. So I think you would have to have a definitive specific antibody test just for COVID-19, and that might be tough because, as you know, we suffer from seasonal coronaviral infections that give us common colds. And so it would have to be a very specific antibody test to COVID. So it's an interesting avenue, worth exploring. But I'm not aware if we have such a specific test available, but it might be possible. You know, as I'm sure you know, there's lots of B-cell epitopes in viral proteins, and you might be able to find one which indicates exposure. I mean, we could use an antibody assay to the spike protein, of course, or we could use an antibody test to the receptor binding domain of COVID. Yeah, I think that's a possibility. You reminded me, I put in a grant to CHR on this, and it wasn't accepted.
Oh dear.
But no, I think, yeah, it's an interesting thought. I think what we would have to do is develop a test to the receptor binding domain, an ELISA test, and show that some people have good titers, equivalent titers to vaccinees. And if we show that, then maybe your idea could be exploited. Yes, yeah, I agree.
Thank you.
Thank you, Catherine. I think Dr. Houghton is going to go to the lab right after this.
Yeah.
He's got another idea. One final comment from the chat box, which I just think is just so fitting to end our time here. This is from a cardiologist. He says, I'm talking from a patient family perspective. Hepatitis C took the life of the most precious person in my life. So the discovery of Dr. Houghton and his team means a lot to me personally. And I thank him very much for his dedication.
Well, thank you so much. And I'm so sorry at your loss. It's actually, it's reminded me that the World Health Organization estimates 400,000 people die of Hep C every year. Obviously, it's not as severe as COVID, but it is still very severe in its consequences. And the WHO estimate there are 2 to 3 million infections of Hep C every year around the world. So thank you for the opportunity to say that that's one of the reasons we badly need a vaccine to Hep C. And once again, my condolences to your great loss.
Thank you, Dr. Houghton. And I want to thank you all here for coming today and for your excellent questions for Dr. Houghton. My special thanks to Dr. Lauren Tyrell for joining the celebration as well. Before we head out though, I just want to draw your attention to several more Nobel Prize events which are coming up next week. On Monday, December the 7th, Dr. Houghton's Nobel Prize lecture will be broadcast in Sweden, and I encourage you to watch UAlberta's social media channels for more details. And then on Wednesday, December the 9th, President Flanagan and I will be hosting a public celebration event in the evening with Dr. Houghton and Dr. Tyrell, moderated by alumnus Dan Riskin. So it promises to be an exciting evening. And then finally on Thursday, December 10th, I hope you'll all tune in for the Nobel Prize ceremony. It'll be broadcast virtually at 4:30 Mountain Time. And last but not least, before we sign off, my gratitude to Dr. Houghton again for his lifelong commitment to advancing virology, and my most sincere congratulations on the Nobel Prize. So please join me once again in a virtual round of applause for our Nobel Prize winner, Dr. Michael Houghton.
Thank you very much, Dr. Hemmelgarn and Dr. Lorne Tyrrell, and thanks to everyone for listening today.
Thank you very much, Michael. And thank you, everyone, and have a good afternoon.